Hello, my name is Quintana-Murci. I work in the Pasteur Institute in Paris, and I'm professor at the Collège de France. And I'm a human population geneticist whose interest in human evolution, how pathogens have shaped the patterns of diversity of the human genome. And also my lab is trying to understand the different factors that drive immune response variation in humans. I remind you that you can submit your questions during this talk via the Q&A function in the top right corner of your screen, and the questions will be addressed later in the live Q&A section at the end of the session. So I would like to thank the organizers of this meeting to give me this great opportunity to share with you this lecture that I guess is a bit an out-of-the-blue lecture for you, because I'm going to talk about evolution, and I'm not going to talk about primary immunodeficiencies. But I hope to convince you by the end of this talk that we can understand immunology, and we can understand also immune diseases by using evolution, probably the most natural experiment ever done. So, traditionally, when we want to understand how genetics impacts on immune diseases, for example, we can use two different approaches. We can use clinical genetic approaches, in which your goal is to try to map a gene and specific mutations that segregate in a family, for example, with primary immunodeficiencies. By doing that, you will, best case scenario, identify a mutation that is highly penetrant, meaning that it explains a large, if not all, the phenotype, and that is rare in the population. On the opposite pole, you have genetic epidemiology studies in which, or GWAS, by genome-wide association studies, in which you are going to compare a population of cases, let's say tuberculosis patients with controls, and again, best case scenario, you are going to identify a few genes, each of them with mutations that are associated with your phenotype, an immune disease, but explain very little of the disease, while these mutations are quite common in the population. So this is not at all neither what I'm going to tell you about today, because we don't work with patients. Actually, we work with healthy donors, people like you and me, the general population, because don't forget that all of us were nothing else than the descendants of those that have resisted past epidemics, tuberculosis, the Spanish flu, the Black Death, etc. So this resistance is somehow engraved in our genes. And this is what we know how to do in population genetics. Just a bit of background, we all originated in Africa around 200,000, 300,000 years ago. And over the last 60,000 years or so, humans have dispersed all over the world. Something that might seem a detail, but it's important in the context of immunity, and I will come later during this talk, is that during these dispersals, we met other human forms that they are now extinct, like Neanderthals in Western Eurasia and Denisovans in Southeast Asia. But during these dispersals around the world, humans have also to adapt to different environments, different climates, different nutrition, and also different pathogens. And this is the basis of human adaptation. Humans, during their migrations around the world, had to adapt to the different pathogens they encountered. So today I'm going to tell you three stories. One about population genetics, meaning how we can use the pressures that have imposed pathogens over time on the human genome to pinpoint immune genes that are of major importance in human survival. I'm going to tell you also an example of a systems immunology studies in which we're trying to dissect the relative contribution of different factors, genetic, environmental, epigenetic, that drive our variation in immune responses. And finally, I will give you two examples of how we can use population genetics to reconstruct the history of past exposure to major infectious disease, malaria in Africa and tuberculosis in Europe. So let's start with population genetics. As you know, natural selection has shaped the patterns of diversity of the human genome. Natural selection, or Darwinian selection, acts on phenotypes, but since many phenotypes are driven by genetics, natural selection leaves signatures, molecular signatures, in our genome. And we have different types of natural selection. Here you have chromosomes, and here that's the past, and that's the present. You have lots of mutations that are in blue, that are neutral mutations. They don't have any impact on any phenotype. But then suddenly you have here kind of dark gray mutations that appear, and these mutations are deleterious, meaning these mutations are associated, for example, to primary immunodeficiencies. So by purifying selection, these mutations will disappear from the population. These mutations, these deleterious mutations, will be purged out of the population. If they are lethal, they immediately disappear in one generation. If they are weakly lethal, they will take a while, but eventually they will disappear from the population. So the genes that evolve under purifying selection are genes that present very little functional variation. In contrast, we have other modes of selection, kind of more positive, like positive selection, in which we go back to the past, lots of blue mutations, neutral, but a red mutation here that appears, and this mutation is not deleterious, but this mutation is beneficial. Let's say, an example, they protect against tuberculosis. So over generations, this mutation will increase in frequency in the population. This is one form of positive selection. And then you have balancing selection, that is a form somehow of positive selection in which the advantage is not the red mutation, but the advantage is being heterozygote, like the iconic case of hemoglobin S that in the heterozygote state protects against falciparum malaria, despite the price to pay is that homozygotes develop sickle cell anemia. But which are the immunological implications of this? Genes evolving under purifying selection, these ones, are genes, as I told you, present very little functional variation. So these genes are involved in essential and non-redundant functions. And since these genes cannot accumulate mutations, when they do, this means that they are going to be involved in severe disorders. And I will come to a minute with some examples. In contrast, genes evolving under positive or balancing selection are genes where novelty or simply diversity is favored. And there are genes with functions that participate in differential human adaptation to pathogen pressures. And we'll see examples of that. Over the last 15 years or so, our main goal has been to dissect the type of selection acting on innate immunity genes in particular, to try to make parallelisms between the type of selection acting on these genes and how essential or redundant are their functions. This is one of the first examples with it in which we studied the patterns of selection in microbial sensors, innate immunity receptors, toll-like receptors, NOD-like receptors, RIG1-like receptors, etc. And what we found here in color code, you have genes evolving under strong purifying selection in red, and genes evolving under neutrality in gray. So What's the immunological implication of that? This means that, for example, among toll-like receptors, you have a dichotomy. Endosomal toll-like receptors are evolving under a strong purifying selection, meaning that they fulfill essential and non-redundant functions with respect to cell surface TLRs, which are more redundant. Let's take the example of toll-like receptor 5. Here, we found a mutation in toll-like receptor 5 that is a stop codon, acts in a dominant negative manner, and is present in Europeans and Middle Easterners from 10 to 30% frequency, meaning that up to 30% of European and Middle Easterners are knockout for TLR5. And at least today, they live very happily. This doesn't mean that TLR5 has a function that is useless, that is, in this case, is the sensing of flagellin, but it means that the function is redundant. In the absence of toll-like receptor 5, you will have other genes that will take over. We've done this at the genome-wide level. We have identified also the 1% of innate immunity genes that are the most under purifying selection, meaning the most essential. And our hypothesis is that these genes, the ones under a strong purifying selection, should be those mutations in which are associated in severe disorders. And clinical genetics of immune functions support this hypothesis, because mutations in some of these genes or the pathways they trigger have been involved in severe life-threatening diseases such as herpes simplex encephalitis, Mendelian susceptibility to mycobacterial diseases, bacterial infections, etc. But we can also adapt. A few years ago, we identified 8 mutations in type 3 interferons that we propose that they have conferred a strong selective advantage to European and Asians. And proof of concept that our methods can identify the right mutations is that some of them, like this one, have been associated with a spontaneous hepatitis C virus clearance. We've done this again at the genome-wide level, and we have identified beneficial mutations in this case, specific for Africans, for Europeans, and for Asians. And our approach is not only telling us, for example, TLR1, 6, and 10 have been beneficial in Europeans, but allow us to specify and to identify the exact mutations that have been the cause of this selective advantage. The next question we addressed here was, well, if we have these selections of mutations in Africans, in Europeans, and in Asians that have conferred selective advantage, when selection started? In population genetics, we can date the time at which these mutations started to be beneficial. And we found that in most cases, selection started between 10,000 and 3,000 years ago, which coincides with the period at which human populations started to shift from being nomadic hunter-gatherers to being farmers and having a sedentary lifestyle, which we all know that this major transition in human lifestyle led to increased population densities, cohabitation with animals, all of which create a kind of perfect cocktail for some pathogens to which humans had to adapt. So to finish with this first part, let me tell you something kind of funny, that is the relation between Neanderthals and immunity genes. So we know that Europeans and Asians, we all have in our genomes around 2-3% of Neanderthal DNA. We know this basically indicates that our ancestors, around 40,000 years ago, had mixed with Neanderthal. That's why today, you and me, of European or Asian origin, we have 2-3% in our genome of Neanderthal material. So we get inspired by the work of Peter Parham from Stanford, that around 10 years ago, he showed that some HLA haplotypes in us, modern humans, comes directly from Neanderthals. So our question here was, well, what about innate immunity genes? Are innate immunity genes enriched in Neanderthal ancestry with respect to the remainder of the genome? Because if the answer is yes, that means that early Europeans, for example, when they had mixed with Neanderthals, they acquired from Neanderthals mutations that allowed them to better resist pathogens, basically. So this is what we did. We examined the levels of Neanderthal ancestry in around 1,500 innate immunity genes, receptors, effectors, signal transducers, and the answer was yes. Both in Europeans and in Asians, innate immunity genes are enriched in Neanderthal ancestry with respect to the remainder of the genome. Here you have an example of the region of toll-like receptor 1, 6, and 10. Each line is an individual. In this case, are around 80 Europeans. In red shadows, you have the genomic position of TLR1, 6, and 10. And in white means that is a normal human haplotype. And in blue, you have a Neanderthal haplotype. So you can see that many of us Europeans, we have TLR1, 6, and 10 covered by a Neanderthal haplotype. We identify around 80 genes in European and 80 genes in Asians, non-overlapping, that present among the highest levels of Neanderthal ancestry of the human genome. Some of them, as you can see here, are involved, like some type 1 interferons, IFITN1 to 3, are involved in antiviral functions. So keeping on Neanderthal, what we also showed a few years ago is that if we focus on mutations that are involved in the regulation of gene expression, these mutations in monocytes are enriched in Neanderthal ancestry, but particularly mutations that regulate antiviral responses. Here, mutations that are associated with responses to TLR7/8 activation and to influenza virus activation of monocytes. Here you have this example of this gene, PNMA1. This gene, the more this gene in red is enriched in Neanderthal ancestry, the more this gene is going to be expressed in human monocytes. And this gene has been involved in the direct recognition of influenza virus. More generally, we wanted to understand how Neanderthals have participated in different modes of the regulation of genes, microRNAs, promoters, enhancers. And to make a long story short, what we found is that enhancers are enriched in Neanderthal ancestry, particularly those that are active in T cells, here in red, and in fat tissues, here in blue. And in particular, we show that they're enriched in T cells that are active in any type, in enhancers that are active in any type of T cells, but not in other cells like NK, B cells, or monocytes. So let me tell you now about our work in systems immunology. Basically, what we try to understand is which are the factors that make us different in terms of our immune responses in health, in the general population. Why some people are here that have a very hyper-responsiveness immune system, or why others are here, are low responders. Is that genetics? Is that environment? Is that ancestry? Is that epigenetics? Is that nutrition? Is that lifestyle? So basically, these are the kind of questions that many labs are trying to address in systems immunology. In our case, what we do is that we have a cohort, usually of different populations. We take their cells, it can be monocytes, it can be PBMCs, it can be whole blood, that we activate either with ligands or with real pathogens. And in our case, we study, for example, gene expression. And then we map the genetic basis of gene expression, therefore of transcriptional responses to immune activation or to infection. And we try to dissect the contribution of genetics versus other factors in our differences in immune responses. So for that, we have been working with two cohorts. One is Evo-immunopop, that is basically individuals of African and European ancestry, all living in Belgium, to homogenize as much as we can environmental exposure. And in that case, we work with monocytes. The other project or cohort is called Milieu Intérieur. In this case, we minimize genetic differences because they are all Western French from Brittany, but we work at the whole blood level. So let me give you an example of both. For Evo-immunopop, as I was telling you, we recruited 200 individuals, Africans and Europeans living in Belgium, from which we purified monocytes, and monocytes were exposed to different TLR ligands, activating TLR1, 2, TLR4, TLR7, 8, but also we used a live strain of flu. We did RNA sequencing to assess the transcriptional responses to these different ligands or virus, and then we mapped the genetic basis of that by EQTL mapping, meaning mutations that are associated with differences in gene expression. As you can see in this PCA, principal component analysis, the main differences are observed between conditions. Here you have the non-stimulated samples, influenza virus infected, TLR4 in red, TLR1, 2 in green, and TLR7, 8 in blue. But if you focus in one of the conditions, like let's take the example of TLR1, 2 responses, you can start to see differences between Africans and Europeans in the way they respond to immune activation. Of course, the differences are not on-off. The differences are in the intensity to which they are going to respond to immune activation or infection. And to make a long story short, that I'm not going to get into the details, what we found is that most of these population differences in immune responses are due to genetics, therefore mutations that control gene expression, and these mutations are just differently distributed between the two populations. In some cases, a single mutation can explain a strong population differences in immune responses. And this is what we call master regulators of gene expression, or trans-EQTL. One trans-EQTL is in the interferon beta-1, that is associated with the expression of many other genes in trans. But let me give you an example of TLR1. A mutation in TLR1 is present, as you can see here, in just 2% of people of African ancestry, but in almost 70% of people of European ancestry. This mutation in TLR1 is associated with a drastic reduction in NF-kappa B activity, but most importantly, this mutation in TLR1 is associated with a strong reduction in expression of around 100 genes involved in inflammatory responses. In other words, the 70% of people of European ancestry that have this mutation have a reduced inflammatory response. Moreover, this mutation has been found to be under strong positive selection in Europeans, suggesting that a temperate, a reduced, a slightly dampened immune response has been beneficial for Europeans. Let me give you now an example of Milieu Intérieur. In this case, the idea was to be a bit more broad in the number of factors that we study that might explain immune response variation. So for that, we recruit 1,000 people in Brittany, but they are stratified by 5 decades of life, from 20 to 70 years old, 5 groups of 200 each, half women, half males. For each of them, we have different types of biological material, nasal swabs, stools to assess the microbiome, but also we obtain whole blood from each of them, and using true culture, each tube was stimulated with different microbes, different activators of the innate and the adaptive immune system. And most importantly, from each individual, we have a 44-page questionnaire about demographic and lifestyle variables. So first of all, we assess And I'm not going to get into the details of that, but both the transcriptional signatures at the whole blood level of each one of these ligands, but also the protein profiles associated with each one of these ligands. But let me share with you two examples. One was this one, in which our question was, which are the different factors that drive cellular heterogeneity in whole blood? So for that, in the 1,000 donors, we define around 130 immune phenotypes by flow cytometry. And then we associated these 130 immune phenotypes, meaning all these different immune cell populations, to 140 different variables: genetics, age, sex, smoking, nutrition, if you rent even or if you buy your house, etc. And basically we found that just 5 variables have a major impact in cellular heterogeneity in whole blood. And these are genetics of the host, CMV seropositivity, age, smoking status, and sex. And interestingly, what we found is that while variation in adaptive cells is mainly driven by non-genetic factors, such as age in green or CMV seropositivity in light green, heterogeneity in innate immune cell populations is mainly driven by host genetic factors that are here represented in blue. But what about, this is at the steady state, whole blood, but what about our immune responses to infection? So for that, what we did was assess in the 1,000 donors the gene expression profiles after infection with a number of bacteria, like E. coli, Staph aureus, BCG, a fungi, Candida albicans, a virus, influenza, and a superantigen. And as you can see here, you can see that the PCA mainly explain the infection condition. But you can start to see that even within a single infection condition, like you can see here for influenza, there is inter-individual variation. Some individuals here in orange behave almost like non-responders, like non-stimulated in gray, and others are hyper-responsiveness. So which are the factors that drive this variation in transcriptional responses to infection? So basically what we found is that the first factor in gray here that explains around 20% of the variance is our natural levels of heterogeneity of cells in whole blood, followed by genetic factors that explain around 10% of the variability, and followed by age and sex. Of course, these are average across the genome because there are some genes, but 70% of the variation are explained by genetic factors, and other genes like STAT1 and STAT2, where most of the variation is explained by sex or age. In any case, still, a large fraction of the variance in immune response variation remains unexplained. And now we are trying to understand how other factors such as epigenetics or collections of rare variants, genetic variants, may explain this extra immune response variation that for the time being, we don't know how to explain. So in the last 10 minutes, let me explain you 2 examples of how population genetics can help us to reconstruct the history of exposure to major infectious diseases. One is malaria. Malaria, as you know, is an iconic example, is one of the major killers in terms of infectious diseases today in the world, together with tuberculosis and HIV. And as you know, it's an iconic case, the hemoglobin gene of balancing selection in humans, because the heterozygote state is associated with protection against falciparum malaria. Is present in around 8% of African populations. So here our rationale was, if malaria didn't exist, because the sickle cell mutation is deleterious in homozygosity, it will disappear in 12 generations. So if we date the age at which selection started to occur at the sickle cell allele, we are dating the time at which malaria started to be a burden for the population, because in the absence of malaria, the sickle cell mutation will disappear. So for that, we study around 500 individuals from different parts of Africa, the hemoglobin gene. And to make a long story short, what we found was that the sickle cell mutation appeared in the ancestors of present-day agriculturalist Bantu speakers in Africa around 25,000 years ago, pushing back the time at which malaria was thought to start to be a burden in Africa. And secondly, what we saw is that hunter-gatherers from the rainforest, so-called pygmies, acquired the sickle cell mutation only over the last 6,000 years. What from the farmers? What does it mean? This is called the poison antidote hypothesis, meaning a population of farmers started to interact with another populations, in this case of hunter-gatherers, and brought malaria with them. But they brought malaria, but at the same time, they brought also the mutation that allowed the pygmy population to resist malaria. Let me finish with the example of tuberculosis, another major killer today. But today, well, thanks to antibiotics and to some extent to the vaccine, in Europe, tuberculosis is less a problem than it used to be, but is documented to be the most lethal infectious disease in the history of Europe. So in that case, we took advantage of a recent work by the laboratory of Jean-Laurent Casanova at the Rockefeller Institute, in which they discover the first common mutation that is associated with clinical TB. Of course, there's many mutations that have been associated with rare cases, but in this case, they found a mutation that is present in between 2 and 3% in Europeans, so therefore is common, and is associated with clinical TB. Watch out, because these pie charts, this is not 25%, this is 2.5%. So as you can see, it's present in populations of European ancestry in between 2-3%, and in populations that present admixture with Europeans, like some American populations. So basically in this case, what we did was to analyze the frequency of this mutation in around 1,000 ancient genomes of Europeans that have lived in the last 10,000 years. And to make a long story short, here you have how the mutation in TYK2 has fluctuated over time over the last 10,000 years of European history. It started to be present in Europeans in the Mesolithic times, increase in frequency to almost 10% during the Bronze Age, and then drastically drop in frequency until the frequencies of today of around 2%. So our analysis have shown that this drastic decrease in frequency is due to negative selection. Basically, what we have shown, just to finish, is that this mutation in TYK2 appeared 30,000 years ago in the ancestors of Europeans, but that just 2,000 years ago, after the Bronze Age, started to decrease in frequency and present one of the selective coefficients strongest of the human genome, meaning that has been strongly deleterious for European health. In other words, this population genetics study suggests that tuberculosis have imposed a strong burden on Europeans over the last 2 millennia. So with that, I would like to thank you, all the real actors of this work, the people in my lab, our great collaborators, including Jean-Laurent Casanova and Laurent Abel, among others, and funding agencies, and thanks to all of you for your attention. Thank you.
Thank you very much, Luis, for a wonderful and inspiring lecture. Very refreshing. There's a few questions from the audience, and one is, let's say, a challenging question, I would say. How do you explain the reduction of toll receptor 1 activity in Europeans and in the same time an increase of autoimmune disease?
Okay, thanks, Isabel. Also, I would like now to thank you officially live for this lovely invitation. And yeah, that's a challenging question because the increase in autoimmune inflammation and allergies in Europe in particular coincides, and there's wonderful epidemiological studies on that, with the decrease in infectious diseases. So actually that's my only interpretation of that is that if it not was, so can you imagine if TLR1 pathway was not dampened, how there will be today the increase in autoimmune diseases? I mean, we do know that this selection for diminished inflammation in Europe, it lasts for at least a few thousand years. So that's the only thing I can answer to that.
Okay. More comments and questions. The one question is interesting. Is there a way to capture both pathogen pressure and non-disease associated variation in larger populations? That is a first question.
So how to capture pathogen pressures and non-disease related variation? Yeah, because, yeah, of course, yeah, now I got it. You can increase in frequency of mutation because this mutation set protects you against tuberculosis, but with the same haplotype, you are also increasing the frequency of linked, slightly deleterious mutations. There's examples of that, of how you can hitchhike, let's say, also slightly deleterious mutations, that they cannot be very deleterious because if not, they would be also purged from the population.
Mm-hmm. Okay, a question from Loïc Dupré. He asked actually, how would you explain that the Neanderthal inherited locus has recently been shown to play a detrimental role on susceptibility to severe COVID-19, and otherwise the Neanderthaler really fit immune system? How do you explain that collision?
Okay, so let's start with the second part of the question. Don't forget that the ancestors of Europeans, when they got into Europe from Africa, they were not adapted to the European environment, the cold environment, the nutritional resources, and the pathogens they found in Europe. But they found another human form, Neanderthals, that they were in Europe for at least 300,000 years. So they were adapted to the environment. So we have shown, as you have seen in the talk, that, for example, we borrowed from Neanderthals alleles that increase our resistance, most likely to viral infections. Now, this recent Nature paper of Svante Paabo shows indeed that this region in chromosome 3 that we did inherit it from Neanderthals is associated with increased not susceptibility to infection, but increased severity of COVID-19, specifically increased risk, 3 times more risk to get hospitalized, right? So this is, again, this kind of trade-offs of nature that things that were advantageous in the past, through a mixture or just because a mutation appeared, and this mutation in the past allowed us to better resist malaria, with changes in lifestyle and environment, are not necessarily the most advantageous today. So the more likely hypothesis and the less convoluted hypothesis to explain this kind of counterintuitive results is that this region in chromosome 3 provided us a selective advantage, and now I'm speculating, probably by increasing the inflammatory response to fight pathogens, that now is creating too much inflammation in these patients, reason by which they develop severe disease. The second part is pure speculation. We have no idea about the functional consequences on this stretch of DNA. And this is one of the things that I am interested in doing in the lab, is basically Neanderthalizing that part of chromosome 3 with CRISPR-Cas9, introducing the Neanderthal mutations and assessing in vitro which are the functional consequences of these Neanderthal mutations at the immune level.
Very interesting. Another question that comes back a few times is the question that is also intriguing to me is how can you comment on or maybe model in a way the impact that modern medical care interventions and public health systems will have on what you're describing on the process of evolution and pressure and positive and negative selection?
Yeah, this is a very interesting question to which I always give the same kind of copy-paste answer. And my only answer for that is, let's set up a research group in 5,000 years, and there will be wonderful population genetic results by showing that something major happened in the history of modern humans in the 20th century, that with the introduction of antibiotics, vaccines, and hygiene, the levels and the intensity of natural selection was completely modified. In regions of the world where we have access to that. Don't forget that in many parts of Africa, South America, and Southeast Asia, natural selection is acting in real life.
Mm-hmm, mm-hmm. So there's another question from Alexander Horschen, who asks, his question is gone. So the clustering, actually the clustering of the different genes, how can you explain that? I mean, the enrichment and the positive pressure on several gene clusters, how can you explain that in another way than purely pathogen-driven?
Sometimes, could it be homeostasis? Mm-hmm. Actually, in the specific case of TLR1, 6, and 10, we show that the mutation that is responsible for the pattern of positive selection in Europe is associated with a drastic reduction in NF-kappa B activity, and more generally, is associated with a reduction of inflammatory responses. Could it be just indirectly pathogen-driven? We don't know. Actually, this question could just be keeping homeostasis, probably. One of the most difficult things in population genetics is to pinpoint the exact pathogen driving the selective pressure. Unless you know, as is the case for a substantial number of genes related to malaria, or the only example I gave about tuberculosis of the mutation that Jean-Laurent Casanova in TIC2 identified that for the moment is the only common mutation associated with clinical forms of TB, then ascribing a specific pathogen to a specific pattern of selection is more storytelling than science. So it's very difficult.
Thank you. Another question pertains to the fact if you can derive the evolution of HLA associations, which may differ across ethnicities and be potential drivers of autoimmunity and immune dysregulation.
Yeah, this is another very interesting question that there's wonderful work by, as we know, Peter Parham, Mary Carrington, all these people working on HLA, and also showing how there is a co-evolution of HLA and the KIRs. Having said that, in population genetics, HLA has been largely overlooked over the last 10 years, basically because of the high complexity of the genomic region. Now, I anticipate that in the next 5 years, HLA will be super fashionable again, because with long risks and Pacific Bioscience, we can completely revisit with NGS techniques the evolutionary history of HLA.
Exciting. So a question from Andy January. Are there similar findings from the Denisovan DNA? Are there indeed stretches of DNA that confer advantage or disadvantage to survival?
Yeah, this is a great question that pleased me enormously because it's something that we are working on the Pacific. As you know, Europeans and Asians, we have 2-3% of our genome that comes from Neanderthal, but in the Southeast Asia and the Pacific, they have an extra 2-3% of Denisovan ancestry, which makes that they have 6-7% of archaic ancestry. Archaic meaning other modern humans, not them being archaic, obviously. So in a study that we have been doing in the South Pacific, in Vanuatu, Santa Cruz, Polynesian, all these populations of near and remote Oceania, we have increasing evidence to suggest that while Neanderthals provided us selective advantage related to different phenotypes, pigmentation, adaptation to cold, and immunity, surprise, Denisova almost exclusively gave us things, good things related to immunity. The paper is under review in this moment, but I can tell you that is very contrasting, apparently, what Neanderthal and Denisova gave us, because Denisova gave the ancestors of present-day Pacific Islanders advantageous variation related to immunity.
Okay. Very exciting. So another question is whether you could give a model or create a model that will help you to derive actually it's a very difficult question. So how helpful is the use of population groups like African and Asian, which stem from largely social constructs? And could you propose a system of analysis that expresses your result as a gradient or spectrum of variation across population without using these categories? But yeah, I suggest you...
Yeah, well, this is a great question because it's just putting the finger in the reality of the things. Of course, what does it mean Europeans? What does it mean Africans? There's hundreds of different Africans, cultures, groups, and genetic clusters. There's hundreds of different Asians. Even Europe, that for a long time I have made the error of saying that as a population, genetics is a very boring continent. It's not. There's many different layers of European migrations that gave rise to present-day Europeans. So the person that is asking this question is perfectly right. And of course, when we're using a group of Europeans, of Africans, of Asians, indeed, in my case, I'm grouping a use of Belgian people, a group of, people from Congo, and we should be more specific when we talk about that. But indeed, the short answer that is starting to be very long, sorry for that, is that in an ideal world, we should take clusters of genetic populations, not of self-identified or culturally identified populations, different clusters in Europe, many different clusters in Africa, because the richness of genetic diversity in Africa is immense. And of course, there's the funding problem there to do this kind of analysis. But that will be the ideal setting.
There was also a question on whether you can derive where, if you can derive it at all, where in evolution there has been actually the advantage of silencing or silencing one of the X chromosomes. Oh. Is that so? Yeah, it has to do probably also with positive and negative pressure. But did you look into that?
I'm not at all a specialist on X inactivation. One of my colleagues in the Collège de France, Edith Hurt, that now is the director of MBL, she's the worldwide specialist on that. She could answer. I don't know.
Not a problem. So another question that popped up is what is the meaning or the significance of the fact that it's especially the endosomal TLRs that are under positive selection, as opposed to the surface toll-like receptors. Is that saying something or telling us something about the pathogens that are involved in these selective pressure actions, or how do you? What is your opinion?
My interpretation of that is double. The fact that the endosomal TLRs are under strong purifying selection, not negative, not positive, sorry.
But not positive.
Purifying selection is either because the sensing of viruses is because they sense, not viruses, but they sense nucleic acids, principally from viruses. They are not sensing flagellin that is only in bacteria. They are sensing something, nucleic acids, that we do have also in ourselves. So probably this kind of status quo of endosomal TLRs being strongly constrained is because they have reached an equilibrium at which they recognize nucleic acids from viruses or some bacteria while not recognizing ourselves. And basically, you cannot alter this system because it's dangerous to do it.
Mm-hmm. All right. So I think most of the questions you have addressed. I think people can contact you if they have further burning questions. I want to, with the audience who are also all congratulating you, I want to once again congratulate you, Luis, for a wonderful lecture, for wonderful research, and for a magnificent presentation. And I want to thank you all for listening. Thank you, Luis, for being here with us for the Q&A. And that's the end of today, at least for the live sessions, but I hope you all have a wonderful evening.
Thank you very much.
Thank you, Luis. Bye-bye.
Bye-bye.